Here is a number the pharmaceutical industry does not advertise. Of the 250 most prescribed drugs in the United States, approximately 60% were derived from or directly inspired by natural compounds, most of them plants. The industry knows this because the industry ran the research. Pharmaceutical companies have maintained ethnobotanical research divisions since the mid-20th century, specifically to study traditional plant medicine for developable compounds. They sent researchers into Appalachia. They cataloged what the granny women used, what the curanderos used, what the Ayurvedic physicians prescribed. They tested those plants in laboratories. They found active compounds. And then, in case after case, they discovered that the plant itself could not be patented because it grew in the ground for free and had been in common use too long to qualify for intellectual property protection. So they did one of two things. They modified the compound slightly, just enough to make it technically novel, filed a patent on the derivative, and sold you the synthetic version at a markup the original plant could never sustain. Or they dropped the research entirely and sold you a different drug for the same condition. The mountain women of Appalachia were using the source plants for both strategies across 2 centuries before any pharmaceutical researcher walked into the hills to look. Before we start, hit subscribe. Every item on this list has a paper trail. Let us count down. Number 20. Passion flower for anxiety and sleeplessness. The Appalachian granny women used passion flower, the spectacular climbing vine of the southern mountain forests, for the nervous exhaustion they called being strung tight. They brewed the dried aerial parts into a tea for sleeplessness, racing thoughts, and chronic anxiety. Pharmaceutical researchers investigating passion flower found that its flavonoid compounds, specifically chrysin and vitexin, bind to the same GABA-A receptors targeted by benzodiazepine drugs, producing anxiolytic and sedative effects through an identical neurological mechanism. A randomized controlled trial published in the Journal of Clinical Pharmacy and Therapeutics in 2001 found passion flower extract equivalent to oxazepam, a benzodiazepine, for generalized anxiety disorder with significantly fewer side effects. The pharmaceutical companies that ran early passion flower research found they could not patent the plant or its primary active flavonoids. They patented synthetic GABA-A modulators instead. The benzodiazepine market currently generates over $4 billion annually in the United States. Passion flower grows wild throughout the southern Appalachian Mountains. Number 19. Skullcap for nervous system support and seizure prevention. Skullcap, the delicate blue-flowered mint family plant growing along Appalachian stream banks, was used for the convulsive conditions mountain people called fits, for chronic headache, and for nervous exhaustion following prolonged illness. Pharmaceutical researchers identified bicolin and bicolin as compounds with confirmed activity at GABA-A receptors and significant neuroprotective effects in laboratory models of seizure and oxidative neurological stress. A 2014 study in phytotherapy research found significant reduction in anxiety scores in human subjects using skullcap without the cognitive impairment associated with pharmaceutical anxiolytics. The compound profile prevented patent filing on the plant itself. Pharmaceutical companies have since patented multiple synthetic analogs of bicolin for neurological applications. The original plant continues to grow along every Appalachian creek bank, unpatented and uninstructed. Number 18. Hawthorn for heart failure and cardiac arrhythmia. The hawthorn tree on the edges of Appalachian fields was used by mountain healers for failing hearts, racing hearts, and the irregular heartbeat mountain people described as the heart skipping. German pharmaceutical researchers ran landmark clinical trials on hawthorn through the 1990s, confirming improved cardiac output, reduced symptoms, and enhanced exercise tolerance in early-stage heart failure patients. Those studies were among the most rigorous ever conducted on a botanical medicine. The active oligomeric proanthocyanidins and flavonoids could not be patented because they occur naturally. German pharmaceutical companies sold standardized hawthorn preparations under European regulatory frameworks. American pharmaceutical companies, operating under an FDA framework that does not provide this pathway, declined to pursue it. The cardiac drug market in the United States generates over $40 billion annually. Hawthorn grows on every farm boundary in the Appalachian Range. Number 17. Witch hazel for inflammation and venous insufficiency. Every American pharmacy stocks witch hazel as a skincare product. What most Americans do not know is that the distilled witch hazel on pharmacy shelves is a diluted shadow of the full-strength preparation Appalachian families made from fresh bark and leaves. Pharmaceutical researchers studied witch hazel tannins for chronic venous insufficiency, the painful condition of inadequate blood return from the legs affecting approximately 30% of Western adults. A 1994 study confirmed significant reduction in leg swelling and heaviness. The active tannins cannot be patented. Pharmaceutical companies developed and patented synthetic vascular drugs for the same condition instead. The full-strength witch hazel preparation was left to the skincare industry, which diluted it, bottled it, and sold it as a toner. The shrub grows on every wooded hillside in the eastern United States. Number 16. Red clover for hormonal balance and menopausal symptoms. Red clover, the common pasture plant covering Appalachian fields, was used by granny women for change-of-life symptoms, the hot flashes, mood disruption, and sleeplessness of menopause. The phytoestrogenic isoflavones in red clover, specifically forminetin and biocanin A, drew intensive pharmaceutical interest in the 1990s as controversy around synthetic hormone replacement therapy generated demand for alternatives. Multiple clinical trials confirmed that red clover isoflavone preparations reduced hot flash frequency and severity. Pharmaceutical researchers filed patents on isolated and concentrated isoflavone preparations, the patentable extracts of a plant they could not patent itself. Products derived from those patents are now sold as pharmaceutical HRT alternatives at significant monthly cost. The red clover growing in every Appalachian pasture contains the same isoflavones in their original matrix. The granny women who used it had no idea they were treating a future pharmaceutical research target. Here is the mechanism that explains every item on this list. A natural plant compound that occurs in nature and has been in common use cannot be patented under United States intellectual property law. The investment required to gain FDA drug approval is between $1 and $2 billion. No pharmaceutical company will invest $1 to $2 billion in a clinical trial for something they cannot then exclusively sell. This means that no matter how strong the evidence for a plant's efficacy, no matter how consistent the traditional use and preliminary research, the clinical trial system that produces FDA-approved drugs will not be used to confirm it unless the plant compound can first be modified enough to qualify for patent protection. The system is not designed to evaluate plants. It is designed to evaluate compounds that someone can own. Every plant on this list fell outside patent protection and therefore outside the clinical investment that would have made the knowledge official. The granny women who used them did not need the knowledge to be official. They needed it to work. Number 15. Burdock root for skin disease and anti-inflammatory support. Burdock, the large-leaved plant with notorious hooked seed burrs, grew in disturbed soils around Appalachian homesteads and was used for chronic skin conditions, liver complaints, and persistent joint inflammation. Pharmaceutical researchers identified arctiginin, a lignin compound in burdock root, as a confirmed inhibitor of NF-κB signaling, the same inflammatory pathway targeted by multiple pharmaceutical anti-inflammatory drugs currently generating billions in annual revenue. A 2011 study demonstrated this mechanism clearly. Arctiginin cannot be patented in its natural form. Synthetic analogs have been filed for patent by multiple pharmaceutical research groups. The burdock root that Appalachian healers dug from the ground behind the house addressed the same inflammatory pathways that pharmaceutical companies are now attempting to target with patentable synthetic compounds derived from its chemistry. Number 14. Stinging nettle for allergy, inflammation, and prostate enlargement. Stinging nettle was used by Appalachian healers for hay fever and respiratory allergies, for joint inflammation, and for the urinary difficulties of aging men that mountain families understood long before the condition had a clinical name. German pharmaceutical researchers in the 1990s ran multiple clinical trials on nettle root extract for benign prostatic hyperplasia, confirming significant improvement in urinary flow and symptom scores comparable to pharmaceutical alpha blockers in several studies. The German regulatory system approved nettle root for this indication. The American pharmaceutical industry, which generates over $3 billion annually from BPH medications, did not pursue the nettle research because the active compounds could not be patented. Your grandfather's generation called it a weed. The pharmaceutical researcher called it a liability. Number 13. Yellow dock for iron deficiency and liver support. Yellow dock Rumex crispus, the tall plant with wavy-edged leaves and rusty seed clusters, was used by granny women for what mountain people called rundown blood, the pallor and fatigue of iron deficiency, and for liver complaints indicating impaired bile function. Research published in the 1990s found that yellow dock preparations significantly enhanced non-heme iron absorption in human subjects, addressing the same deficiency the granny women were treating. The active compounds are not patentable. The iron supplement market generates over $800 million annually in the United States. The plant that enhances iron absorption better than many supplement formulations grows as a weed in every agricultural field in America. Number 12. Horehound for bronchitis and productive cough. White horehound was the granny woman's standard treatment for the wet productive coughs of bronchitis endemic in communities of miners and woodsmen. The leaves and flowers were brewed into bitter teas or made into syrups with honey. Horehound contains merubian, a bicyclic diterpene lactone with confirmed expectorant and mucolytic properties, and is among the best documented botanical expectorants in the pharmacological literature. The FDA reviewed horehound as an expectorant ingredient in 1989 and removed it from over-the-counter cough preparations, not because the evidence of its efficacy was negative, but because the evidence base did not meet updated standards. The pharmaceutical expectorant market, selling the synthetic alternative guaifenesin, generates over $1 billion annually. Horehound grows wild across the Appalachian range. Number 11. Red root for lymphatic congestion and immune support. Ceanothus americanus, called red root for the color of its inner bark, was used by Appalachian healers for swollen lymph nodes, spleen complaints, and states of lowered resistance following prolonged infection. During the Civil War, it was documented to have anticoagulant and immune-stimulating properties noted in field journals on both sides. Red root contains ceanothic acid and alkaloids with confirmed effects on lymphocyte production and fibrinolytic activity. Patent barriers prevented commercial development of the natural compound. Synthetic fibrinolytic drugs derived from the same mechanistic research generate billions annually in hospital settings. The plant that Appalachian families used as a spring lymphatic tonic grows on dry, sunny hillsides throughout the eastern United States. Here is the number that should make every person who has filled a pharmaceutical prescription uncomfortable. A 2019 research analysis of all new FDA-approved drugs between 1981 and 2019 found that 66% of all new small molecule drugs were either natural products, natural product derivatives, or synthetics directly inspired by natural product structures. More than half of all cancer drugs approved in that period were natural product derivatives. The pharmaceutical industry's public position on plant medicine is that it is insufficiently studied and potentially dangerous. The pharmaceutical industry's private research record is that it has been systematically mining plant medicine for lead compounds, modifying those compounds enough to patent them, and selling the modified versions at prices the original plants could never support for the entire history of the modern drug industry. Number 10. Mayapple and the chemotherapy connection. Mayapple Podophyllum peltatum, the umbrella-leaved plant carpeting the Appalachian forest floor every spring, produces a root that granny women and indigenous healers use topically for wart and abnormal skin lesion removal. Pharmaceutical researchers studying mayapple isolated podophyllitoxin from the root, a compound with remarkable cytotoxic activity against rapidly dividing cells. They could not use podophyllitoxin directly as a drug because its toxicity was too broad, so they modified it. The synthetic derivative etoposide, a direct chemical modification of the natural podophyllitoxin in mayapple root, became one of the most widely used chemotherapy drugs in the world and remains standard of care for small cell lung cancer, testicular cancer, and certain lymphomas. The plant cannot be patented. The modified derivative generates hundreds of millions of dollars annually. Every Appalachian family that knew mayapple would remove a skin lesion was using a plant whose chemistry was modified into a life-saving cancer drug without acknowledgement or compensation. Number 9. Trillium for postpartum hemorrhage and women's health. Trillium erectum, the striking three-petaled wildflower called birthroot, was used specifically for excessive menstrual bleeding, uterine prolapse, and the postpartum hemorrhage that represented fatal risk in mountain communities without surgical access. Trillium contains steroidal saponins and phytosterols with confirmed hormonal and uterotonic activity studied by pharmaceutical researchers investigating phytohormones for gynecological applications. The compounds are not patentable in their natural form. The pharmaceutical gynecology market, selling synthetic uterotonic and hormonal products, generates tens of billions annually worldwide. Trillium blooms every spring in the same mountain forest where granny women gathered it, unpatented and unreimbursed. Number 8. American beautyberry is insect repellent and anti-inflammatory agent. American beautyberry, Calicarpa americana, the shrub producing striking clusters of iridescent purple berries along Appalachian fence lines, was used in mountain communities for a specific practical purpose. Its crushed leaves and twigs rubbed on the skin repelled insects. Before chemical repellents, in communities where tick-borne illness and mosquito-borne malaria were genuine threats, this was a medical tool. The USDA Agricultural Research Service investigated beautyberry in the 2000s and identified colicarpinol, intermediol, and spathalinol as primary active compounds. Controlled laboratory testing found colicarpinol comparable to DEET in deterring mosquitoes and biting insects. The compounds cannot be patented because they occur naturally and have documented prior use. The commercial insect repellent market generates over $1 billion annually. The shrub producing the relevant compounds grows in every hedgerow from Virginia to Texas. Here is the regulatory architecture that locks this system in place. The Dietary Supplement Health and Education Act of 1994 created a framework in which natural plant products cannot be marketed with disease treatment claims regardless of how strong the evidence is. A manufacturer of passionflower capsules cannot state that passionflower reduces anxiety, even if a randomized controlled trial confirms it performs equivalently to a benzodiazepine, because the FDA has not approved passionflower for that indication. The FDA will not approve passionflower for that indication because no pharmaceutical company will fund the billion-dollar trial necessary when the result cannot be patented. The plant sits in regulatory limbo, confirmed by research but officially unrecognized, while the pharmaceutical company that studied it sells patented synthetic analogs as drugs. The framework was written in 1994 when the supplement industry lobbied for it. The pharmaceutical industry has found the same framework protects them from natural competition. Both industries benefit from a system in which the plant cannot make a claim and the drug can charge any price it chooses. Number 7. Wood betony for headache and nervous system complaints. Wood betony Stachys botanica, a mint family plant brought to Appalachian communities by Scots Irish settlers, was used by mountain healers for persistent headaches, nervous tension accompanying hard physical labor, the headaches of high blood pressure and general nervous system exhaustion. The alkaloids it contains, including stachydrine and botanicin, have demonstrated cholinergic activity and blood pressure-lowering effects in laboratory studies. Pharmaceutical interest in cholinergic compounds has been intense in the context of Alzheimer's disease research. The specific compounds in wood betony that produce cholinergic effects are structurally similar to compounds patented for neurological applications. The similarity is not coincidental. The source plant grows wild and costs nothing to obtain. Number 6. Devil's walking stick for pain relief and anti-inflammatory support. Devil's walking stick, Aralia spinosa, the fearsome thorn shrub of Appalachian hillsides, was used by mountain healers for toothache, joint pain, and the deep inflammatory conditions of rheumatism. The inner bark applied topically produced local analgesia. Aralia species contains saponins and triterpene compounds with confirmed anti-inflammatory and analgesic properties structurally similar to the steroid backbone underlying most pharmaceutical anti-inflammatory drugs. The saponins themselves are not patentable. Synthetic derivatives mimicking their anti-inflammatory mechanism while qualifying for patent protection are the subject of ongoing pharmaceutical research. The mountain people who reached through the thorns to strip this bark were accessing an anti-inflammatory chemistry pharmaceutical research is now attempting to replicate in a form that can be owned. Number 5. Joe Pye weed for kidney stones and urinary infection. Joe Pye weed, Eutrochium purpureum, rising above every wet Appalachian meadow in late summer, was used specifically for kidney complaints, the painful passage of kidney stones, the burning of urinary tract infections, and the deep flank pain of kidney inflammation. The plant contains euperin and other compounds with confirmed diuretic and anti-inflammatory properties affecting the urinary tract. Pharmaceutical research on euperin in the 1980s identified potential applications in urinary pathology. The compound could not be patented. The pharmaceutical urological drug market generates billions annually from synthetic alpha blockers for stone passage and antibiotics for infection. The plant that Appalachian healers use for the same conditions blooms in every wet ditch in the eastern United States every August. Number 4. Goldenrod for upper respiratory infection and kidney support. Goldenrod has been blamed for decades of American hay fever because it blooms at the same time as ragweed, which is actually responsible for the allergy. This misidentification cost goldenrod its reputation in American popular culture and helped pharmaceutical allergy medicine avoid a natural competitor. Appalachian granny women used goldenrod for upper respiratory infections and kidney and urinary complaints. Goldenrod contains quercetin, rutin, and saponins with confirmed anti-inflammatory and diuretic properties. European phytomedicine has used goldenrod as an approved treatment for urinary tract inflammation for decades. American regulatory practice classifies it as a dietary supplement that cannot claim to treat anything. The allergy drug industry that benefited from goldenrod's misidentification has not rushed to correct the record. Number 3. Wild indigo for immune stimulation and infection support. Wild indigo, Baptisia tinctoria, growing on the dry acid soils of Appalachian hillsides, was used by granny women and the indigenous peoples before them for the persistent respiratory infections following initial illness and the states of diminished resistance that left patients vulnerable to successive illness. Wild indigo was studied extensively by German pharmaceutical researchers in the 1980s and 1990s in the context of immune research alongside echinacea. The polysaccharides and alkaloids in wild indigo demonstrated immune-stimulating activity comparable to echinacea in several studies. The compounds could not be patented. The immune supplement market sells echinacea preparations, many containing wild indigo as a secondary ingredient at significant commercial scale, while acknowledgement of wild indigo's active contribution remains substantially less prominent than the echinacea branding. Number 2. Persimmon root for dysentery and antimicrobial wound care. The American persimmon, Diospyros virginiana, produces a root bark that Appalachian families used for the severe dysentery that killed children in communities with contaminated water. The astringent tannins contracted inflamed intestinal tissue, reduced fluid loss, and provided antimicrobial activity against bacteria responsible for diarrheal illness. The same preparations were used topically for infected wounds. Persimmon tannins have been studied in pharmaceutical research for wound care and gut inflammation applications, with confirmed activity against several bacterial species, including E. coli strains responsible for diarrheal illness. The tannins cannot be patented. Pharmaceutical anti-diarrheal drugs generating over $1 billion annually in the United States address the same gut pathology through synthetic chemistry. The persimmon tree produces its medicine every autumn in any fence line or forest margin in the eastern mountains. And number 1, the bill you pay for the modification. Every item on this list follows the same arc. A plant is used by mountain healers for generations. A pharmaceutical researcher identifies the active compound. The compound cannot be patented. One of two things happens. The researcher modifies the compound enough to file a patent, runs the clinical trials the natural compound will never receive, gets FDA approval, and sells you the modified version at a price the plant cannot compete with. Or the researcher drops the plant and develops a synthetic drug for the same condition, leaving the plant in regulatory limbo while the drug occupies the approved indication. The mayapple producing the backbone chemistry of etoposide, a chemotherapy drug, grows in the Appalachian forest for free. The drug costs thousands of dollars per treatment course. The passion flower binding to the same brain receptors as a benzodiazepine grows wild in the southern mountains. The benzodiazepine market generates $4 billion annually. The hawthorn confirmed by German researchers to improve cardiac output in early heart failure grows on every farm boundary in the Appalachian range. The cardiac drugs treating the same indication generate $40 billion. In every case, the patent is not on the discovery of what the plant does. The mountain women already knew what the plant did. The patent is on the modification that makes the knowledge ownable. Here is my challenge. The next time you fill a prescription, look up the drug's mechanism of action and what natural compound the mechanism was first identified in. The answer, in a surprising proportion of cases, is a plant with documented traditional use. Sometimes a plant with documented Appalachian use. The pharmaceutical company did not discover what you are paying for. They found out who knew it first, modified it enough to own it, and sent you the bill. The mountain women never sent anyone a bill. That was always the problem.